My name is Brian Feely. I'm a shoulder and sports medicine surgeon at the University of California, San Francisco. I was fortunate to work with a group of my colleagues to publish our paper in JBJS entitled Sex-Based Differences in Cell Types and Gene Expression Within the Anterior Cruciate Ligament. We are motivated to do this study because sex-based differences in ACL injury are one of the most consistent and frustrating problems we see in sports medicine. We've all heard the data. Female athletes tear their ACLs at least 2 to 8 times higher than males, and even when you control for sport, level of play, and injury mechanism. While biomechanical and hormonal factors have been studied extensively, particularly knot shape, shape of the femur, Q angles, they haven't really fully explained this disparity. What's particularly interesting is the fact that re-injury rates, tears to the graft, happen equally in men and women. So if it was actually due to entirely intrinsic factors, this wouldn't be the case, and women should still have higher failure rates. So we wondered whether part of the answer might be intrinsic to the ligament itself, specifically whether male and female ACLs differ at the cellular level in ways that may affect tissue strength, repair, and ACL degeneration. And to answer that, we used single-cell RNA sequencing. Traditionally, when you study ligament tissue or any other tissue, you grind everything together and get an average signal across all the different cells. Single-cell RNA sequencing lets you do the opposite. It allows us to look at thousands of individual cells, one at a time, look at the genes in a non-biased manner, so very different than PCR, and see which genes it's expressing. Now, this approach let us identify distinct cell types within the ACL and understand how specific populations behave differently in males and females. We did this in 9 patients, which sounds like a small sample size, but it gets us more than 12,000 cells, each with a full non-biased panel of gene expression. Now, one of the most striking and unexpected findings was how heterogeneous the cells in the ACL really are. We identified multiple different distinct cell populations, including fibroblasts, endothelial cells, parasites, and immune cells, all present in pretty similar numbers in both sexes. What surprised us most, however, was that every major cell type showed sex-based differences in gene expression, despite having similar overall proportions. So it wasn't necessarily the cell types that were different, but the gene expression within each cell type that was different between males and females. Now, in particular, we identified a ligament progenitor-like fibroblast population marked by the gene TPP3 in female patients. And in these patients, these cells express genes that are associated a little bit more with collagen dysregulation and extracellular matrix degradation. In contrast, the same cell population males express genes associated with a more robust collagen organization, as well as stem cell maintenance. Now, this suggests that biologic differences in ligament cell behavior may predispose female ACLs to reduce tissue resilience and robustness over time. So maybe there's these repeated micro injuries that don't heal quite as well in female athletes than male athletes. Now, the most important unanswered question is causality. We identified sex-specific gene expression programs, but we don't yet know which of these differences directly drive ligament failure and which are either adaptive or compensatory. And this study isn't really designed to do that. Our next steps are to functionally test these pathways, particularly in the TPP3 progenitor population, and explore whether modulating collagen remodeling or progenitor cell behavior can strengthen ligament tissue and reduce injury risk, particularly in female athletes, but it would be generalizable to all athletes. Now, ultimately, our goal is to translate these cell insights into strategies that help prevent or at least reduce ACL injuries and improve long-term outcomes for athletes of all sexes.